Okay, we already got our oral presentations off the list. Woo-hoo. Now we're going to finish chapter 8. We're going to go from there into looking at our Lab 5 results for what we started on Tuesday, which is a lot of fun. And then we're going to do a preview of Lab 6 because there's actually a couple of different things you need to know. So we'll cover that as well. So we need to talk now about how we use our DNA. That is sort of known as gene expression, and it involves two processes that you've likely heard of: transcription and translation. So what is transcription? We do transcription. What do we mean by that word? Say again? Yeah, so it is a part of that process of taking the DNA sequence and turning it into a protein. And the word transcribe is literally like a letter-for-letter copying of the DNA. So the A, G, C's, and T's in DNA get turned into letters in RNA, which are very similar, except for what? Instead of A, G, C, T, what do we have? A, G, C, U. Okay, so we need to talk about how we do this process of both transcription, which is turning the DNA into a RNA molecule, and then translation is when you take the RNA, which is the letter-by-letter copy of the DNA, and turn it into protein. And it's called translation because it's turning a code into something else, right? We're not copying the letters anymore. Now we're reading them and understanding what they mean. And how is that code related to the protein? How many letters in the code means something? 3 letters, every 3 letters is called a codon, right? And what does that codon tell us? What amino acid goes in that particular position? So it's really most helpful, I think, to watch this in a movie, which is what we will do now.
This is what Francis Crick called the central dogma of modern biology: how DNA makes protein.
Ah. Ow. Go away.
It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching way to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell.
Why the sound effects? I have no idea.
Then, in a dazzling display of choreography, all the components of another molecular machine.
What's going to happen now? the other molecular machine that's going to read the RNA, turning it into protein. What's it called?
Locked together around the RNA to form a miniature factory called a ribosome.
And that, remember we talked about how the ribosome has two pieces? The small and large piece float around independently until they find an RNA. The small piece grabs the RNA first, and then the big piece comes and sits down on top of it.
It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA.
Okay, so when you do that, we'll just let it think about itself for a second. When you do that, what the ribosome is doing is it's putting amino acids together, right? What chemistry is it using to do that? How do we put subunits together in the cell? Remember when we talked about this in chemistry? We use a chemical reaction to put things together and the opposite one to take them apart again, right? So what is that chemical reaction? Condensation, right? Or dehydration, because in order to put a new bond there, you have to rip off the oxygen or OH group hydroxyl group on this side and a hydrogen over here. Those guys get stuck together and then a new bond is formed. There we go. So when we looked at that process, DNA is making messenger RNA that goes to the ribosome. Is messenger RNA the only RNA that gets made? Are there other kinds of RNA? Yes, there are, right? We've heard about ribosomal RNA and transfer RNA. And in that movie, you saw the transfer RNAs with the green molecules. They're just RNAs that don't need to be made into anything to function. They're already good on their own. When we talked about enzymes in the chemistry section, we talked about ribozymes that have this same ability. So in addition to messenger RNA, there are regions of the DNA that code for ribosomal RNA, which doesn't get made into protein, it's just functional already, and it gets put into the ribosome. The ribosome is this giant machine that's made of a lot of pieces. Some of the pieces are proteins and some are RNAs. And then the transfer RNA that you saw in that movie is just made, again, it's just made as an RNA, and the RNA molecule folds into something that already functions. So we know from the last class the difference between RNA and DNA. I'm just going to point it out again that they're very similar. All of the nucleotides in both nucleic acids have a phosphate group. They have 5-carbon sugar and a base, but the 5-carbon sugar in DNA has one less hydroxyl group than the one in RNA, which is the reason for the name. Deoxyribonucleic acid has deoxyribosugar, which was missing a hydroxyl group. And ribonucleic acid, or RNA, has both hydroxyl groups intact. So in order to do transcription, we have to use this enzyme called RNA polymerase. And remember we talked about how DNA and RNA polymerase
basically are just saying you're making a polymer of DNA or you're making a polymer of RNA. If you were doing different kinds of chemistry, then you would have different names for some of these things. So for instance, in the RNA virus that we just heard about, the West Nile virus, in order to replicate the genome, you have to make more RNA. And you're starting with an RNA, right? So how does that work? A lot of the things that viruses do is by co-opting cellular machinery. But if they're in a eukaryotic cell, is there a way for that cell to take an RNA template and make an RNA product? That cell, does it have enzymes to do that? Do we do that in our cells? The answer is no. Okay, our RNA polymerase can only make RNA from a DNA template. So viruses have their own enzymes to do this, and in some cases, we'll see how that works, but in some cases, they actually have to carry the enzyme in the viral capsid. All right, so when we do transcription, what has to happen is that the machine that's going to transcribe the RNA, which is the RNA polymerase, has to bind at a special location just next to the gene. That region is called the promoter, and it's just a sequence of nucleotides, just like all the other parts of the DNA. But the sequence is like a recognition site for that enzyme to sit down. It knows what the promoter sequence looks like. And it's only going to transcribe a certain amount of DNA, right? It's going to page 87 of the instruction manual, and it's only going to copy page 87. It doesn't keep going to page 88 and page 89, because at the end of page 87, there's a little sequence of DNA that's basically a signal to stop transcribing. So this picture kind of just shows you the process. And I won't go through it in excruciating detail except to point out again that there is a sequence of letters in the DNA that are the site, they're like the landing pad for the RNA polymerase. There's a sequence of letters at the end of the gene that are the stop transcribing code. And what the RNA polymerase does is as it moves along, after it sits down on the promoter and begins transcribing, it briefly melts the strands of DNA and copies one of the strands. And the way that we diagram this, just like we diagram replication, is that we always have the 5' to 3' strand on the top as the coding strand. And so what is actually happening is that the RNA polymerase is reading the bottom strand and adding in nucleotides that are complementary to the bottom strand so that the RNA looks like the top strand. Does everybody follow that? Okay, it's reading this, which could be like CCTA, and it's copying by putting in complementary. So complementary to C would be G, G, A, whatever I just said. And that ends up looking like the top strand. And what you can see in this diagram is, here again, the polymerase is reading the bottom strand, it's making a complementary copy to the top strand, and so it's actually this RNA molecule that's being made looks like this DNA molecule in sequence, except for that where there would have been a T, there's now a U. So if we were to look at this using an electron micrograph, as shown in this picture, what you can see is that they've pseudo-colored the DNA strand in blue, and the RNA that's coming off of it in pink. And two things that you can see in this picture are, first of all, you can tell which direction the polymerase is going. Which direction is it going in? It's going to the right. And we know that because as these RNA polymerases move along, this RNA product gets longer and longer. Now, the other thing that this shows you that I think is really important to understand is it's not that the RNA polymerase reads this whole thing and makes a copy, then sits around and has a copy with his friends, and then goes back and does it again. It's not slow, right? What's happening right now? How many RNA polymerases are working at the very same time, would you say, in this picture, if you were to guess? How many RNA polymerases are bound to DNA right now in that particular gene? I would say probably 100. So transcription's happening, like there was a polymerase that bound here about 5 minutes ago that was zooming along, making a copy, and now it's over here. So it's making one of these long strands.
And then right behind him was another guy that's currently making this one. And right behind him was another guy that's making that one, and so on and on until we have also these guys here that are making new RNA strands, and this guy's making one just ahead of him. So it's very, there's a lot of activity. When you transcribe a gene, you don't do it halfway, you just go for broke. So what happens when you do translation? And again, the mRNA is translated in codons, 3 nucleotides at a time. The start of the gene is always encoded by this AUG sequence. Now, AUG is both the start codon, and what that means is that is the sequence in the RNA the ribosome is looking for, and it's also, it codes for methionine. The ribosome knows when to stop because it reads one of these 3 codes, either UAA, UAG, or UGA. So we'll just look at what that looks like in a second, but there are 64 arrangements of letters that you may find in messenger RNA, and there are only 20 amino acids. So what does that tell you? There's more than one way to encode every amino acid, which is why we call the code degenerate, or another way to say it is redundant. Now, the other important thing to know is that the transfer RNA knows which codon to bind to, because it's actually carrying, it's made of RNA. And 3 of the letters of that RNA are the complement to the codon. And the 3 letters in the transfer RNA are called the anticodon. We'll get to a picture that helps you understand that in a second. This is the genetic code, so this chart tells you everything you need to know about how to translate RNA into protein. And if I was to ask you a question on the exam, which is extremely likely, I would provide this code. I don't expect you to just memorize it. And on the code, you can see clearly there's one way to start the process, and there's 3 ways to end it. All right, so let's look at this in detail. So here is a messenger RNA, and there's every little bar represents a nucleotide. So there's some letter here. They've actually spelled out the AUG, but there's a variety that could be an AA, CC, GA, C, U, whatever, okay? There's a couple things to note about this. First of all, on purpose, they've diagrammed extra nucleotides upstream of the AUG site. So when you transcribe the RNA, there's often stuff on the end you're not going to end up using, okay? So this floats around. This little subunit, which in the eukaryotic cell would be a 40S subunit, binds the RNA first, then the big subunit gloms on as well, and they scan this message until they find this sequence. The first sequence they find that's an AUG sequence is where they start. Okay, so it's all put together, and this sequence is going to match this transfer molecule, which is made of RNA. And again, all along here there are A, G, C's, and U's. They just haven't bothered to diagram them. But these 3 are the anticodons, and the anticodon letters. So UAC is the anticodon for AUG, because these match to each other. And it's bringing in methionine, and this is the one that always starts. The next thing that happens is that the ribosome reads the next 3 letters, and the next transfer RNA comes in. The catalytic machinery, the enzymatic activity of the ribosome is to take this amino acid and covalently bind it to that one using dehydration synthesis. And you just keep going until you hit a sequence that has no equivalent transfer molecule. And so if it sits here long enough, then the ribosome just gives up. It releases the polypeptide, the protein, and then the 2 subunits come apart again. And what happens to this message after it's being read? It depends, okay? Messenger RNA is not super stable, but most likely it's read again by another ribosome, and usually another ribosome doesn't bother to wait patiently until this whole thing has been read. Just like the transcription event, you get another ribosome right away, and maybe many, many of them. There's something really important to understand about prokaryotic genetics that's different from eukaryotic genetics that you've learned before. And that is this. You can have transcription and translation happening at the same time in the prokaryotic world. So I'm going to describe it,
And then we're going to talk about why that's happening. In this picture here, this really faint line that you can barely see is the DNA. Now coming off of it is an RNA transcript. In this particular picture, this is where the promoter is, right? And we have about 8 RNA polymerases that are making RNA that's dangling off of this DNA. But there's also all these large round structures that are grabbing onto the RNA. What are those guys? Those are ribosomes. The ribosomes have begun translating this messenger RNA before it's even finished being transcribed. So that means that if you need to make some enzyme in your prokaryote, you can make tons of it really fast, which is great because that's really the only thing that you have going for you. In order to adapt to the world, it's not like you can put in an overcoat when it gets cold or run away from a predator. You have only this ability that you can react very quickly and change your proteome. Now, why is this possible and why is this not happening in a eukaryotic cell? Why does this happen in prokaryotes? Where are these processes happening? Where does transcription happen in a eukaryotic cell? The nucleus, right? Yeah, because we're making RNA out of DNA, so we have to be where the DNA is, which is in the nucleus. Now, where does translation happen in a eukaryotic cell? In the cytoplasm. So there's a barrier, right? You have to make your RNA, finish making your RNA, it has to leave and go into the cytoplasm, and then the ribosomes can find it. That's not true in a prokaryote, right? The DNA and the RNA polymerase and then the ribosomes are all in the same molecular compartment. So you can have this situation where you have a really, really rapid response to things. There's a difference between prokaryotes and eukaryotes, something that eukaryotes do that prokaryotes don't. And that is that prokaryotic genes are simpler. Okay, so the prokaryotic gene, usually the structure is, there's a promoter. That's where what sits down at the promoter. What enzyme goes there? What is the enzyme that's going to actually do the transcription, make the RNA? RNA polymerase, right? So it sits down on the promoter. That's what it's looking for. And then all of the sequence after that is just 3-letter codes for different amino acids until the terminator. In a eukaryote, what happens is that the DNA sequence actually there's a promoter region, and then the coding region has exons and introns and exons and introns. And what does that mean? This exon is the region of the RNA that's going to be kept in the final mature message, and the intron is going to be cut out. So the first thing you do when you do transcription is here's the promoter, here's the sequence of the DNA. So you just copy it, okay? You just make an RNA transcript. This is an immature RNA transcript. Then, because there are certain signal sequences here, another series of enzymes goes in and chops out part of the message and splices these different pieces together. Okay, so that your final mature messenger RNA is shorter and is missing some of the pieces that was actually in the DNA. That's what goes out into the nucleus. So given that that's the case, it's actually really important that you can't have transcription and translation happening at the same time. The fact that you have to be in the nucleus and finish transcription first before you can begin making a protein is really helpful. Why is it that there are these weirdo introns that are there and then you have to get rid of them? That seems really dumb, right? Well, what this helps you with in terms of evolution is that if this protein, say this region has a particular shape and function, and this region has a different shape and function, and this one has a different shape and function, then you can actually, for instance, make a couple different versions of the protein from this sequence. You could make this version where you just chop out all the introns and put all 3 things together. But sometimes the way that this is processed is you chop out this whole section here, then you just put these 2 pieces together, and that actually makes a different protein. It means that with less DNA, you can make more different things. Okay, Now, what group of microbes would we have to worry about this? Are there microbes that are eukaryotes? Yes, absolutely, right? Protozoans, algae, fungi that are microscopic, the microscopic life cycle of the some of the parasites. So here's just a summary of what we've talked about, the major differences in these processes when you compare eukaryotes and prokaryotes. So the presence of the nucleus is different, obviously, in eukaryotes, no nucleus in prokaryotes. How replication happens is different in these two cell types. So we've covered that in the previous class, but basically replication in eukaryotes has to be completed before you can go through cell division. Whereas in the prokaryotes, remember that each cell division can happen more rapidly than it takes to copy the whole chromosome, and the daughter cells can inherit a partially replicated chromosome. And transcription and translation are a little different. So in eukaryotes, transcription happens in the nucleus, and then there's this extra editing step on the message, whereas in prokaryotes, it happens in the cytoplasm. And consequently, translation in eukaryotes is separated from transcription, but in the prokaryotic world, you can have them happening at the same time. And finally, as I have mentioned before, the ribosome is slightly bigger in eukaryotes. So what does this mean in terms of speed? Which one of these cell types is going to be able to go from DNA to finished protein product faster? Prokaryotes, exactly. Okay, so now we have to talk about regulation. And we're not going to cover this exhaustively, but let's ask this question again. Are all the genes in a particular organism, are they all on, which means are they all being made into protein all the time? No, because that's wasteful and we don't need them all. We may have made a ton of this enzyme and there's plenty of it. We don't need to make more right now. Or we might have an enzyme that metabolizes something that's not in the diet right now. So what would be the point? So here's an example. This is an E. coli. Look at this cute little chromosome with a no nucleus. And this is a zoomed up image of just a part of the plasma membrane. And it's importing lactose, which we know is a disaccharide sugar. One piece of lactose is glucose. So when you have the right enzyme, you can chop this disaccharide apart and get glucose and lactose. And glucose can go directly into what process to start making energy? Glycolysis. Galactose has to be done, something else has to happen to that. So because of that, this is not an ideal food source because only about half the molecule can be immediately used for energy. So you're not going to bother to use this if there's just free glucose floating around. If there's free glucose floating around, you're going to import that first and use it up. And you're only going to use this if it's all you have available. There are special enzymes that are required for lactose metabolism. One of them is beta-galactosidase that chops the disaccharide. And so that enzyme's only made if A, lactose is around, and B, glucose is not around, outside in the environment. Now, when we're talking about regulation, there's a couple of terms. Constitutive genes means those that are on all the time, and there are some of them that you do need to use very often because they're, things that keep you alive, basically. And they're just constantly needed. But most of them either fall into one of these camps, okay, either repressible or inducible. So what's the difference? Repressible means that they're on most of the time, but you could turn them off if you needed to. And inducible means that they're usually not on, but if there's some unusual circumstance, you could turn them on. And we'll also talk about this term metabolite repression in a second. I've already talked about most of these terms, so I've already defined what a gene is. That's the instructions for making a particular protein. And that has both a promoter, which is this non-coding regulatory region, and the coding region, right, which is the instructions for making the protein. The coding sequence is that part of the RNA that's actually going to be turned into protein by the ribosome. And this is a new term, operon. Now, in prokaryotes, and pretty much unique to them, genes are often organized into operons. So what does that mean and what does it look like? we're used to sort of, hopefully at this point, we're used to sort of thinking of a gene as being this DNA sequence. And maybe we understand that over here is a sequence that, here's the coding sequence that's actually going to be made into a RNA that's going to be made into a protein. And over here is potentially a promoter, which is not made into RNA. It's just a signal for the RNA polymerase to sit down. But in prokaryotes, usually there's a promoter region here. That's where the RNA polymerase is going to sit down. There is another regulatory region called the operator. We'll talk about what sits down there in a second. And then here is where the coding region is. And it contains multiple genes. So it's coding for gene A, gene B, and gene C that might be different enzymes that are all co-regulated. Okay, so why would you do that? Can you think of a reason? How would that help you? I'll give you an example. Let's suppose that we have something like lactose. In order to metabolize it, we need an enzyme A that's going to chop it into two pieces, glucose and galactose. And then another enzyme potentially is the transporter that brings in the lactose from outside in the environment. And maybe there's a third enzyme that does something else. I don't remember what else it does. But the point is that there's some sort of series of events that has to happen all at the same time. If thing A is true, lactose is around, there's no glucose, then we need all of this stuff made all at the same time. So you may as well have all of the genes you need together in the same place so that when you turn on this program of, oh, lactose is around, we make everything that we need. Now, I mentioned this operator, which is a non-coding region of the DNA, and it is the regulatory sequence near the promoter. That is bound by the repressor. And what is the repressor? The repressor is this protein that sits down on the operator. It's usually encoded over here, and it makes a protein that binds here and stops the RNA polymerase from getting through. So that is a way to regulate whether or not this operon is being made into protein or not. So let's look at an example. So here's a typical operon, and what you can recognize are, this is the promoter sequence. Again, this is shown as a, sort of a stick diagram, but these are letters of the DNA. Next to that's the operator, or letters of the DNA, and this just depicts that it is a place where some protein can bind. More letters that encode all of these enzymes that are needed for some metabolic process. And over here is this gene I, sometimes it's called R, sometimes it's called I, repressor or inhibitor. And when it's made into RNA, it makes this molecule. And sometimes the molecule that's made, the protein, is active and sometimes it's inactive, and we'll talk about those details. But when it's an active repressor, it fits into this operator, and the RNA polymerase, which is on the promoter, cannot transcribe. So you can regulate whether or not there's transcription of this and whether these enzymes are being made by deciding whether or not this repressor is in the way. So again, there are these two terms you need to understand, repressible and inducible. So repressible is for an operon that is making proteins that you need to be on most of the time. So it's going to be on by default. Unless you do something, transcription is happening. And you're only going to stop transcribing when you don't need it. And so an example is the tryptophan operon. What is tryptophan? Does anybody know? What group of chemicals does it belong to? We're going to actually talk about it today because of our metabolic testing, and I mentioned it on Tuesday. It is an amino acid. Yes, ding ding, points to the lady in the front. So, tryptophan is an amino acid that you need pretty much all the time, right? Because it's one of the amino acids in proteins. So anytime there's a sequence in the DNA that's transcribed into RNA that's read by the ribosome, that's the code for tryptophan, you're going to need a tryptophan. So you need a ton of tryptophan. And there are a variety of enzymes that start with the precursor to tryptophan, some reagent up here, and a bunch of enzymes that end up making tryptophan at the bottom as the product. Under what circumstances would you not want to be making more tryptophan? If you have a ton of tryptophan already, right? So this metabolic process has been working over time, and just so happens you didn't need very much of it because your proteins coincidentally didn't have very much of it in them for whatever reason. And so you want to turn this off in those rare circumstances where you have a stockpile of it already. So what would be really convenient is if this was able to go back to the first enzyme in the pathway. So let's look at that enzyme. We'll make a picture out of it. And let's suppose that tryptophan can fit in here in this. Here's the active site. This is where the actual enzyme does work. And over here is just a place where tryptophan can fit. And when it does so, it changes the active site. So if you remember from chemistry, that's called non-competitive inhibition and also called feedback inhibition when the end product of a pathway feeds back on the first enzyme in the pathway. Okay, so tryptophan basically could do that. Now what's actually happening is that tryptophan fits into this protein and turns it from an inactive repressor into an active repressor. And instead of doing this feedback on the pathway, what's actually happening is this active repressor goes away to the DNA. Here's the DNA sequence, right, where all of the enzymes you need for tryptophan metabolism are in a row next to the operator, next to the promoter, next to where the repressor is actually made. And RNA polymerase is sitting here just churning away until this guy that's been activated by tryptophan sits down in the operator and blocks him. Okay? Does that make sense? So only in this rare situation where you have lots of tryptophan are we going to get this repressor working. Normally the repressor is made and it looks like that. You bind tryptophan to it and it sits and prevents transcription. The reason that that's better than feedback inhibition is that it means you don't have to even have to put the energy into making more enzymes. You're not even having to stop the pathway at that level. You're just not even calling the guys into work in the factory in the first place. Okay, so let's talk about this other kind of operon, the inducible kind. So an inducible kind means that you have to actually do something to turn it on, right? And that would be something that you only need sometimes under some circumstances, such as if you were going to metabolize an unusual carbon source like lactose that's not great and you wouldn't normally use unless it was all you had. So I'm going to show you some examples of that. So here's the tryptophan operon. And again, this is what it looks like. So here are this coding region that codes for all 5 enzymes that you need in the tryptophan metabolism pathway. Next to a sequence of DNA, that's the operator where a repressor could sit, but usually doesn't. Next to the promoter, who sits here? RNA polymerase. This is a sequence that's coding for the actual repressor protein that's inactive. So transcription comes along and makes all of this stuff, okay? It makes this guy, and it makes all of these. These are made into all of the proteins that you need to metabolize, synthesize tryptophan. This guy's made into a repressor that is not active, and it just sits around waiting for some kind of signal. Which is, if you have too much tryptophan fits into this protein, turning it into an active repressor, which can now fit into the operator site and prevent polymerase from making more of these enzymes that we don't need right now. On the other hand,
Just the opposite is true in the inducible operon situation. So again, this is the example that we use is the lac operon. This is the operon that has all the genes for metabolizing lactose. So LAC-A, LAC-Y, and LAC-Z. So there's the operator region where a repressor usually sits, the promoter region where the polymerase sits, and then this regulatory gene which is going to be able to make a protein that sits here. So we only turn this on in some circumstances, and what has to happen is that we make this protein and it's active right away. Okay, so unlike the other repressor in the tryptophan situation, this is already able to bind the operator, and it just sits there and prevents transcription unless something happens. The something that happens is a metabolic variant of lactose. Basically, when you bring lactose into the cell, some of it gets turned into this allolactose. That binds this repressor protein, making it inactive. Now it can't bind here, so RNA polymerase binds here. And then finally, we make the different enzymes we need for lactose metabolism. And so that explains in some ways why it is that you have something called metabolite repression, which is that if you're providing a microbe that can do lactose metabolism and glucose metabolism with those sugars, then what you'll see is that there's sort of a rapid increase in cell numbers. This is rapid growth. Then a lag time where nobody's growing, and then a slower increase. And what's happening is that in this period of time, we have exponential growth because we have ideal conditions and lots of glucose. And glucose gives us the most energy compared to lactose. Then we run out of glucose, and there's this lag time. It's as if we just started a new culture. There's no more glucose. The lactoses are still around. It hasn't been used up. But you have to actually take a little bit of time to make the enzymes you need to metabolize lactose before you switch to lactose metabolism, which is going to give you less energy and your growth gets slower. Okay, so this just summarizes those two circumstances, basically. I'm going to take a short break at this time, and then when we come back let's come back at 5 minutes after 7, and we are going to talk about mutation and then also horizontal gene transmission.